All right, we're going to jump into our next unit, which is about cells, and in particular about cell membranes. And the reason we are focusing on cell membranes, when you were first learning about cells, maybe in middle school or whenever that might have been, you probably learned about a cell membrane that was this outer part of a cell, and then there were all these cell parts on the inside, a nucleus, a mitochondria, and whatever. And you learned about the cell membrane, which was this thing on the outside. Well, in fact, certainly that is true. The cell membrane is the barrier that makes the cell a structure at all. It separates this cell from its outside environment. But in fact, all of these organelles on the inside, this nucleus, this mitochondria, a Golgi apparatus, whatever you might be talking about, all of these are simply made out of membrane as well. They are little bubbles, little sacs made out of membrane in the cell. So cell membranes aren't just those things that contain all the guts of the cell, if you will, this outer part. Cell membranes make up almost all the important parts of the cell. And if we're going to learn how cells function, not just from for how all their parts function, we need to understand how cell membranes function, because they make up not just this outer part, but all these inner parts as well. They are very, very important fundamental structures. The fundamental building block of the cell is the cell membrane, at least of the eukaryotic cell. So we need to learn about cell membranes. The cell membrane separates the cell from its outside environment, first and foremost. If we're just talking about this outer membrane, indeed, if we talk about the organelles, of course, those membranes that create the organelles separate the interior of the organelle from the cellular environment, but be that as it may. Regardless of what location we're talking about for a particular membrane, it cannot be an impenetrable boundary. It certainly is a boundary separating in from out, but it cannot be impenetrable. The cell needs things from its environment. Things need to get in, and things need to leave the cell. For example, you can imagine food, all the macromolecules that we learned about in our biochemistry unit, oxygen, and so on, as well. Things need to get out. Waste products like nitrogenous waste, ammonia, we'll talk more about that later on, carbon dioxide, even if certain useful products, your cells in your salivary glands are creating amylase to break down starch, if you'll recall that. And so that's a product your cells are making. It can't stay trapped in your cells. It has to be allowed out. So products need to get out as well. So this idea of selective permeability, membranes are selectively permeable or semi-permeable, meaning they are barriers that are going to allow some substances across them, but not all substances. This is extraordinarily key. You need to recognize these terms, selective permeability or semi-permeability. Okay. Now, fundamental structures of cell membranes, they are composed of lipids. We need to go back to our biochemistry unit just a little bit. And remember, lipids and proteins, these two molecule categories make up cell membranes. Lipids are the foundational molecules. They are the fundamental bricks, if you will, in the cell membranes. Phospholipids, if you'll recall, we talked about these. They phospholipids are made up with that phosphate group. Well, let me go back. If we remember that a lipid looks like the letter E, and these three are fatty acids, we're going to replace one of those with this phosphate group. So phospholipids, and proteins are like windows embedded in the brick wall, if you will, of the phospholipid brick wall. Okay? And those, remember, proteins are the things that do actual work in our cells. So the proteins that are actually embedded in the cell membrane are what are going to be doing work for that cell membrane. The lipids are important. They're the foundational structures, the bricks in the wall, but the proteins are what actually allow the cell membrane to do things. Okay? Proteins do all the work. Okay, that's important to remember. Okay, so you need to recognize these diagrams. Phospholipids are usually in shorthand drawn to look like this in most texts and diagram form, with the circle being the phosphate head, if you will. One end of the phospholipid, that end with the phosphate group, and then these two lines down here represent the fatty acid tails, as they're called. This is a more technical drawing over here showing you how the molecules would more really look. Obviously, this is quite simplified over here. The phosphate head, if you'll remember, a phosphate group roughly is a phosphate with some oxygens all around, and those oxygens are negatively charged. Therefore, it is quite polar or attractive to water. It is hydrophilic, this phosphate head. It is attracted to water. The fatty acid tails, being hydrocarbons, just carbon, hydrogen, are quite nonpolar. Water is not interested in bonding with those fatty acid tails, so it is those fatty acid tails are hydrophobic. Water ignores them, pushes them away, and as a result of how this thing responds to water, it's got this split personality, if you will, this water-loving part over here and this water-fearing part down here, and because of that, when placed in water, phospholipids will water will arrange them like this, where the blue here indicates water, and water loves to cling to these phosphate heads. Water loves to cling to these phosphate heads, but just by default pushes the tails away and towards each other. And so you arrive at this bilayer structure, a two-layer structure of phospholipids, and this is the fundamental structure of all cell membranes, this phospholipid bilayer structure. And again, structure relates to function. The structure of these phospholipids relates to how they come together to form these cell membranes that make up our cells. Okay? And this is what we just talked about. Okay. Now, because of the way the phospholipids cling to each other and their polar nature, this sets up this idea of selective permeability, that there are some things that can get in through this bilayer and some things that cannot. So, if we've got this bilayer, let's say here it is, there's our phospholipid one. Okay, there's our bilayer. Some things can pass through it. Some things hit these phospholipids, however, and cannot pass through it, and they get stuck here, if you will. So, what can penetrate is anything that basically is not polar. If it's not polar, these polar heads will not be interested in sticking to it, and so it can kind of just slip between those phospholipids. Lipid molecules, fats, oils, being nonpolar, can do that. Oxygen, remember, O2 is nonpolar. It's oxygen clinging to itself has an equal pull on electrons. It is nonpolar, so it can get through. Carbon dioxide is a nonpolar substance. It can get through just by moving in between phospholipids because they're nonpolar. They don't get attracted and stuck to the polar heads. However, anything that is polar is not going to pass through. Ions are going to immediately be attracted or repelled from those phosphate heads. Water will be attracted to them and stuck. Glucose is a polar carbohydrate, reasonably large molecule. It's not going to get through. Any amino acids that are polar. So these molecules are going to either be too large, too polar. They're going to attract water molecules to them that are going to make them even larger. If you'll remember the talk about hydration shells around certain ions. So they are going to get stuck and be trapped outside next to these polar heads, and they will not be allowed to pass. There's a hydration shell right there showing the water molecules around sodium and chloride, for example. So, wrapping up, polar part, phosphate heads. All of this to the interior is the hydrophobic tails, okay? Okay, and they serve as the barrier or border that is going to allow only certain things to pass in and out. Now, lipids can pass, okay, nonpolar. What else can pass? Well, sugar cannot. It's going to get stuck next to those polar heads because it is polar. Water cannot. It is stuck to those polar heads. Salts, because they are ion, they will be broken down into ions, cannot. Waste is also polar. It cannot escape. You might notice that this sets up some certain problems. We need waste to get out. We need sugar to get in. We need water to be able to get in. So how then can these polar but molecules that need to move get through the membrane? And that's where we need to remember that proteins are also fundamentally important to cell membrane structure. These are the windows in the brick wall. These are how we get certain molecules in and out that cannot pass through the phospholipids themselves. These proteins are embedded in the bilayer, like chips in a chocolate chip cookie, okay? Proteins can do any number of different things when embedded inside a membrane, but many proteins act as channels, or little tunnels, if you will, through which molecules can scurry through and get from one side to the other of a protein. So, remembering that proteins can be very, very, very specific in how they function. Certain specific channels are going to allow certain specific materials, molecules across the cell membrane, and they will not allow others. So again, protein structure is very specific to function. One protein may allow one type of molecule to pass and not another. So, for example, that protein might have allowed that sugar molecule to pass, but this one will only allow a certain amino acid to pass. This one might only allow water to pass. This one might only allow salt to pass through. This one might only allow waste to pass through. That's ammonia. So, each different type of protein and channel embedded in the membrane will be responsible for one particular type of molecule passing. Okay. Now, This is an important special channel that we're going to actually identify its name. This particular protein channel is called aquaporin, and you ought to be able to recognize it. Obviously, given its name, you probably can figure out what it does. Aquaporin is the channel responsible for allowing water to pass through a cell membrane. It used to be thought that water didn't need a specific channel, but then it was discovered not too many years ago. Nearly all cell membranes have aquaporin molecules embedded in them in extremely large amounts. Because water is polar, obviously, it would get hung up around the phosphate heads, so we give it a little door through which it can get through, which is represented by this yellow ribbon-looking structure here. That's representing the aquaporin, and we can watch the molecules just go through from an area of high concentration to low concentration, and we'll talk about that later on. Okay, so aquaporin allows water molecules to move with their concentration gradient from one side of a cell membrane to another. Okay. Okay, so we've got that, I think. Okay. This is a view showing you just how these protein channels would be embedded in this bilayer. They are embedded all the way through. Obviously, if one of them, for example, is going to act as a tunnel, perhaps, we'll give it a little opening and a little tunnel in the middle here for molecules to pass through. It's got to be embedded all the way across the membrane. And so most of these proteins in a membrane are referred to as transmembrane proteins because they cross the entire membrane. And proteins truly are the perfect molecule to do this. And why that is, if you'll remember, proteins are made of amino acids. Some amino acids are nonpolar in nature, and some of them, some of them are polar. Okay? These are polar ones. These are electrically charged. And they are going to, because these amino acids have different characteristics in and of themselves when they come together to make a protein, some regions of the proteins that are going to become embedded in cell membranes will have perhaps nonpolar amino acids arranged in this region, whereas they will have polar amino acids arranged out towards the outer parts of their structures so that they can embed and lock into this cell membrane. So within the membrane bilayer, you will find nonpolar amino acids that are going to anchor that protein into this nonpolar region of the phospholipid, but on the outer surfaces or the outside regions right here, the amino acids making up this transmembrane protein will be polar, and so hydrophilic, and so they will behave just like these polar heads here, and they will lock themselves into place that way. So there's some aquaporin right there showing it allowing water to pass through, and this region of this molecule would be polar, allowing it to lock into the membrane, and this would as well, but not so much so that the water cannot pass through the tunnel. And we'll talk about that later. Okay, so membranes do a lot of different jobs. They act as transport channels, as we've seen, like right there. They act simply as enzymes, just catalyzing certain chemical reactions, not necessarily letting anything pass through. They will act as receptors for certain molecules. We'll talk about this more, not just letting this molecule pass through, but receiving a molecule on the outside and then allowing that to cause something to happen on the inside. They can act as identity markers on cells. This particular protein right here, all the protein parts are in purple, has an identifying carbohydrate attached to it, which can identify a cell as belonging to a particular individual or a particular organ or whatever, and for immune system purposes, and we'll talk about that later. Certain proteins can allow cells to actually stick to each other, or they can allow be anchoring points for cytoskeleton fibers that act as a support structure for the cell to be put in place. So fundamentally, you need to understand that proteins can do a lot of different things when in cell membranes. Okay? Depending on what type of proteins are embedded in a particular cell membrane determines what kind of job that cell membrane might be doing, and thus what kind of job that cell might be doing. Okay? And there are several types of proteins, which we just saw, but there's another way to talk about them. We can also talk about them as peripheral, that are kind of just sort of loosely stuck to the outer part of a membrane, or we can talk about integral proteins, which are more of this transmembrane protein type. Integral means it's an actual foundational part of the cell membrane. So integral would be talking more about transmembrane proteins, like we talked about earlier, those that go all the way across the membrane. So if you see these two terms, peripheral and integral, this means kind of stuck onto the surface. Integral means embedded in. That's all that's getting at. Okay? All right. We need to make sure you understand this phrase. Fluid mosaic model is simply the term for this idea that the cell membranes are made up of bunches of little parts. If you know what a mosaic is, it is a picture made up of many small tiles. Well, that's how you need to think of a cell membrane. It's made up of a lot of little pieces, but it is fluid. It's not stuck in a stationary way. These little pieces can flow past one another. Okay? And that brings us back to this element. We learned about cholesterol, and you still need to remember what it fundamentally looks like, these 4 carbon rings with various things hanging off of them. Cholesterol has a really important job in maintaining cell membrane structure. Okay? What you're seeing here are phospholipids, and this is showing you how cholesterol would be embedded in between them and helping the cell membrane maintain its structure. You need to think of it as almost like a type of cellular antifreeze. Antifreeze in your car keeps your car from freezing up, the liquids in your car from freezing up in the winter, but it keeps them from boiling over in the summer. It keeps the membrane, cholesterol keeps the membrane firm but still fluid. In cold, it's not going to let it freeze up. In hot, it's not going to let it become so liquid that it loses its integrity, that it loses its structure. Okay? So though we often think of cholesterol as being bad, it is bad when it's in the wrong places, in your blood vessels, clogging things up, for example. But it's when it's in its cells, the way it's supposed to be, it's not only not bad, but it's actually quite necessary. Okay? So, you should understand this diagram showing how a cell membrane is put together. This is illustrating the fluid mosaic model. These molecules would be able to kind of slide around each other. They're not static. They're not going to flip-flop a lot, but they can slide around each other. And you are seeing exoskeleton. This would be the inside where the cytoplasm, the guts of the cell are. This is the outside of the cell. And what we're seeing here are cytoskeletal support structures that are attached to the cell membrane and helping to support it. There's your cholesterol. Transmembrane proteins, a peripheral protein that is just kind of stuck to the outside or to the surface of the membrane. And that is called a glycoprotein. It is called glyco. Glyco always refers to having a carbohydrate, so this is a protein with the carbohydrates. This is probably a cell-identifying protein structure, and that's a glycolipid. Again, probably perhaps used in identifying the cell as a particular type of cell, but it is a lipid, but instead of the phosphate head, it's got a carbohydrate. So again, glyco means carbohydrate, but lipid tells us it's attached to a lipid. Okay? This is a little bit of detail on cell-to-cell recognition or cell-identifying molecules. These carbohydrates are important in identifying cells as belonging to you and not some foreign invader, and so on. So we'll talk more about this as we get moving on into some other organ systems like your some other body systems later on. That's it for cell membrane structure. We've still got a lot to talk about with cell membranes, but we'll pack that up in another podcast. Thanks a lot for watching. I'll see you in class.